Welcome to another deep dive today. We're going to be exploring a pretty cool method for detecting testosterone in water.
Sounds interesting.
Yeah, it is. We're going to be looking at this 2013 research paper. Let me get the title here. Okay. A testosterone-specific competitive enzyme immunoassay for monitoring water quality.
Catchy.
Right. It was published in the Bulletin of Environmental Contamination and Toxicology. And the authors, Chachaporn Yaripong, Victor Wong, and Nanju Alice Lee, they're from the University of New South Wales in Australia.
Okay.
And they detail their development of a pretty clever new technique for detecting that testosterone in water. And they use something called an ELISA, or ELISAR.
Okay, so let's unpack this a bit.
Yeah, let's dive in. Maybe we can start with you. Why is detecting testosterone in water even important? I mean, it doesn't seem like the most obvious concern at first glance.
Well, you're right. It's not something people typically think about, but testosterone is actually an endocrine disrupting compound, or EDC for short.
EDC.
And even at low concentrations, it can disrupt the hormonal balance of aquatic life and, impacting reproductive cycles and overall health.
Makes sense.
Plus, there's growing concern about potential long-term effects of EDCs on human health.
So how do scientists measure testosterone in water now? And what are the issues with the current methods?
Well, the traditional methods involve techniques like chromatography and mass spectrometry.
Right.
But they need expensive equipment, specialized expertise, and they're really time-consuming.
I see.
So widespread monitoring is really a challenge, especially for places with limited resources.
So that's where this ELISA comes in.
Exactly.
So what is an ELISA and how does it work? You said ELISAR, right?
Yeah, ELISAR. It stands for Enzyme-Linked Immunosorbent Assay.
Okay.
In simple terms, it uses the binding interaction between an antibody and its target.
The target being testosterone in this case.
Right. These researchers, they designed a modified testosterone molecule, and that let them make antibodies that are very specific to testosterone.
So it's like a lock and key system. The antibody is the lock and testosterone is the key.
Exactly. Now, in a water sample, you've got all sorts of stuff,
Yeah.
including maybe some testosterone.
Okay.
These researchers, they introduce a known amount of an enzyme-conjugated testosterone molecule.
Okay.
This conjugated molecule, it competes with the testosterone that's in the water sample for those antibodies.
For the binding sites.
Right, for binding to those antibodies. So the more testosterone in the sample, the less of the conjugated testosterone can bind.
Okay, so how do they measure how much testosterone is actually there?
This is where the enzyme part comes in. The
Yeah.
enzyme that's attached to the conjugated testosterone that triggers a color reaction when you add a specific substrate. And the intensity of the color, it tells you how much testosterone is there.
Okay.
Darker color means less testosterone. Lighter color, more testosterone.
So they can indirectly measure it. Interesting. Pretty clever.
It is. And what's really impressive is how sensitive this ELISA is. They found a limit of detection, it's also called IC20, of 0.05 plus or minus 0.01 micrograms per liter.
Wow, okay. That's really sensitive. Can you maybe put that into perspective for our listeners?
Sure. It's basically like finding a single grain of sugar in an Olympic-sized swimming pool.
Okay, yeah. That really drives it home.
Yeah.
So sensitive. What about, I know there's a term, cross-reactivity.
Right.
Did they test for that?
Yeah, they did. They wanted to make sure the ELISA only detects testosterone and not other similar molecules.
So it's not picking up,
Yeah.
things that are similar in structure.
Exactly.
And did it pass the test?
Oh, yeah. They found less than 0.05% cross-reactivity with other steroids. That's a big win.
It is.
Yeah.
So we've got a sensitive and specific ELISA for detecting testosterone. But I imagine it's not all smooth sailing when you take this, out of the lab and into the real world.
Yeah, you're right. The real world is messy.
It is. What did the researchers find?
All sorts of things can affect the accuracy, like pH levels, certain metal ions, especially copper, and even organic matter. Things like humic acid, they can all interfere.
So it seems like even those small things can really throw a wrench in the works.
You got it. So careful sample preparation is key here.
To getting good results.
Absolutely. You have to remove or neutralize those substances that interfere.
So it's not just about the ELISA itself. It's about understanding the water samples, right?
Exactly. They call it a matrix effect in the paper,
Yeah.
meaning all the stuff in the sample besides the testosterone that can affect the results.
Gotcha. So it sounds like there's a bit of an art to sample preparation to make sure you're getting a clear reading.
I like that. A clear reading.
So with a well-prepared sample, did this ELISA do well? when they tested it outside the lab?
That's a great question. Well, to evaluate the ELISA's performance in real-world situations, Chachaporn Yurapong, Victor Wong, and Nanchu Alice Lee, they took 50 surface water samples.
50! Wow. Okay, and where did those come from?
They were all from New South Wales, Australia,
Right.
but from a bunch of different places: rivers, lakes, wastewater treatment plants, even agricultural runoff.
So they really wanted to see how this ELISA would hold up.
Right, and they wanted to validate their findings. So they compared the ELISA results to a well-established technique,
Okay, which one?
the yeast-based androgen screen,
Okay.
the YSS assay.
So like comparing it to the tried and true method.
Exactly. And the ELISA did great. There's a strong correlation between the ELISA and the YS results. So it suggests that this ELISA can measure testosterone levels in all these different water samples reliably.
Even those complicated ones you mentioned.
Yeah, even the ones with more complex compositions.
Wow. Okay.
Oh.
So that seems like a big point in favor of the ELISA.
Right. It is. But they also did spike and recovery tests.
Spike in recovery.
Yeah, it means they add a known amount of testosterone to the samples and then see if the ELISA can detect it.
So like a test within a test.
You could say that. For the cleaner water samples, like the river water and the seawater, the recovery rates were great. It was accurate.
So even with all that other stuff in the water,
Yeah.
the ELISA could pick out the testosterone.
Exactly. But for the trickier samples, like the wetland samples and the tile drainage water, the recoveries were less consistent.
So in those more complex samples, it was harder to get an accurate measurement.
Yeah, that's what they found. And they say it highlights the need for more optimized sample preparation. You have to get rid of those interfering substances before you use the ELISA.
Especially with those more challenging samples.
Right.
So there's still work to be done.
To get the most out of this ELISA, yeah.
Yeah.
But overall, this research is a huge step forward, for monitoring endocrine-disrupting compounds.
Definitely. This has been so interesting. I mean, we've seen how this ELISA works, how it performs in the real world, and it seems like there are so many possibilities with this research.
Yeah, it's really exciting, not just for monitoring testosterone, but for other emerging pollutants, too.
I mean, if we could adapt this for other contaminants, that would be a game changer.
Absolutely. Early detection of these things. That's so important if we want to assess the risks and do something about them.
So it's about having better tools to diagnose those problems. Right?
Exactly. It's like having a more refined toolkit for protecting our water.
This ELISA could really transform how we monitor the environment, I think.
Yeah, I agree.
This has been a fascinating look at this really complex topic. It's amazing how much goes into, something as seemingly straightforward as measuring testosterone in water.
You're telling me. It's been great talking about it.
Thanks for walking us through it all.
My pleasure.
So we've talked about the science behind this ELISA, and we've seen how it works in real-world water samples. But what does all this mean, for the bigger picture, for environmental monitoring?
Well, this ELISA, it could really change things. The traditional methods, they need all that fancy equipment. It's expensive.
Right.
So it's hard to do a lot of monitoring.
Especially like we were saying before, in those areas that don't have a lot of resources.
Exactly. But this new ELISA, it's faster, simpler, cheaper.
So we could be monitoring testosterone levels more often. And in more places.
Yeah. Think about it. Smaller labs, environmental organizations, even just local communities, they could all be monitoring their water.
And see if there are any risks from these, what were they called?
EDCs,
Huh.
endocrine disrupting compounds.
Right, the EDCs. So it's like giving people more control over their own water.
That's a great way to put it. And it's not just about testosterone.
Right, because if they can make an ELISA for testosterone,
They can make them for other contaminants, too.
that's pretty amazing.
It is. Think about it. Pesticides, pharmaceuticals, all that industrial waste. If we could detect those things early, it would make a huge difference.
It would. For risk assessment and for taking action.
Exactly.
It's like having, a better way to check the health of our water systems.
I like that.
So we can, treat them before things get worse.
Yeah. Early intervention is key.
This ELISA, it really feels like a big step forward in protecting our water.
It is. It really shows what science can do when we need to solve these environmental problems.
Well, this has been a fascinating deep dive. I've learned so much about this ELISA and what it can do. Thank you for explaining it all.
It's been my pleasure.
And to all our listeners, remember that these kinds of scientific advancements, they're crucial for protecting our environment. Stay curious, stay informed, and let's all work together to keep our water safe and clean.